Hi everyone, I am Dr. Sheela, Assistant Professor, PG and Research Department of Microbiology, St.Joseph Arts and Science College, Autonomous, Cuddalore. Today discuss about the topic mass production of bacterial biofertilizer. First of all, what is biofertilizer? A biofertilizer is a substances which contain living microorganism. When applied to seed, plants, plant surface or soil, the biofertilizer add nutrients through the natural process of nitrogen fixation, phosphate, zinc, potassium solubilization, and stimulate the plant growth through the synthesis of plant growth substances. Isolation of bacterial biofertilizer. First one is isolation of rhizobium. The approved leguminous plants that test for nodule presence. The wash the root system is running tap water. Color of nodules vary from white brown to pink green. The undamaged pink color nodules are selected and washed again with the distilled water. They keep selected nodules in 0.1% of acidified potassium chloride solution for 5 minutes to remove the contaminant from the surface nodule. Repeatedly wash the sterile water to wash out the disinfectant. The immersed the ethyl alcohol to sterile the root nodules after wash the sterile water. The root nodules as are crushed and sieved through the fine sieve. Then the extract is serial diluted and inoculated the culture medium. The selective media for rhizobium, EMA medium, yeast extract mannitol agar medium is used for the isolation of rhizobium. The culture plate are incubated for 10 days at a 25 degrees Celsius. After 10 days, the rhizobium will be grown on plate. Next one is isolation of phosphate, zinc and potassium solubilizing bacteria. The soil sample, 10 gram of soil sample is transferred to 100 ml of sterile distilled water and mixed thoroughly by shaking the flask for few minutes. The serially diluted of soil suspension is made using sterile distilled water. The 1 ml of sample from appropriate dilution up to 10 power 6 is spread even over solidified cooled Pickovskia media for phosphate solubilization, tris mannitol agar media for zinc solubilization. Next, Alexandra media for potassium solubilization. Alexandra media for potassium solubilizing. After incubation at 28 degrees Celsius in incubator for 3 days, 3 to 7 days, colony appear. The clear zone around the bacterial growth indicate the bacterial and zinc and potassium solubilization. The commonly used nutrient solubilizing bacteria is Pseudomonas and it's isolated for following steps mentioned below. This one is isolation of bacterial biofertilizers. Isolation of Pseudomonas. The root are washed in tap water to remove the adhering soil particles using sterile knife, blade or etc. The roots cut into small bits of 1.0 centimeter in size. Using the sterile forceps, slightly crush root bits in sterile water and serial dilution of root suspension are made using sterile distilled water. And 1 ml of suspension is spread evenly over solidifying cooled Kingsby media in Petri plate. The plates are incubated at 28 degrees Celsius for few days and fluorescent bacterial colony will appear on surface of Kingsby media. The colony showing the fluorescent pigment under UV light are isolated for further work. Next one is mass production of biofertilizer. The mass production technology of biofertilizer It is used for some beneficial bacteria such as Rhizobium, Azospirillum, Acetobacter, Phosphobacteria, Pseudomonas have been standardized. The bacterial biofertilizer production technology involved the following steps: preparation of starter culture from mother culture, production of culture broth in fermenter, preparation of carrier material, mixing culture broth with the carrier and curing, package and storage. This one is mass cultivation of bacterial biofertilizer. First one is to prepare the mother culture and a slant prepare and prepare the broth and prepare the broth in small flask and a big flask prepare the culture and then shaker for limited requirements and then the quality checking for culture. The broth is blended with a sterilized carrier peat or lignite soil, packing in polyethylene bag. First one is preparation of starter culture from mother culture. The mother culture is obtained from microbiology lab or culture collection center to the gram staining to check the contamination. Take 100 ml of liquid media in 250 ml of conical flask and steriled using autoclave. The transfer loop full of inoculum into liquid media. Keep the flask in shaking condition around 30 degrees Celsius for incubation. Check the population whether it reaches 10 power 10 to 10 power 11 colony forming unit. The incubate until the maximum population is reached. The culture obtained is this way is called as starter culture. Some important beneficial bacteria under broth under pH range. Rhizobium, the beneficial media, yeast extract, mannitol broth. The pH ranges from 6.5 to 7.0. Acetobacter, waxman base media 77. pH 6.55 to 7.0. Azospirillum, malate broth with a NH4Cl 6.8 to 7.0. Gluconeostobacter, LG broth, pH ranges from 5.5. Pseudomonas kingsby media 7.2. Phosphobacteria, nutrient broth, pH ranges from 5 to 7.0. Production of culture broth in fermenter. Fermenter is a vessel which maintains the controlled environmental condition for the growth of biofertilizer organism. Fermenter is made up of stainless steel to withstand high pressure and avoid erosion. The fermenter provide access for inoculation sample, aeration and cleaning. The volume of fermenter may vary from 10 liter to 5000 liter. Generally, culture broth of biofertilizer production is prepared in fermenter of 10 to 5000 liter capacity and sterilized in fermenter vessel itself by passing steam generated from sterilization unit 120 to 130 degrees Celsius. The pH ranges should be maintained using some types of chemical NaOH and HCl. The continuous aeration should be given by the part of fermenter is called as sparger. They incubate the culture broth with fermentation process. What are the incubation period? 3 to 7 days. The preparation of carrier material. The medium which contains a bacterial biofertilizer allowed to multiply ideal carrier material should have the following properties: very cheaper, locally available, high organic content, contain non-toxic chemical, 50% of water holding capacity, easy to process, buffering capacity. The carrier materials commonly used for peat soil, lignite, vermiculate, charcoal, pressmud, farmyard manure, coconut husk, rice husk, fly ash. Next one is mixing culture broth with the carrier material and curing. The culture broth possesses a minimum population of 1 billion cells per ml of blended with the carrier. The final moisture of the product should around 40 to 50 percentage. After mixing, a blended carrier is kept for 24 hours for curing in cool temperature. During this time, the bacteria get assimilated with the carrier materials. Okay, thank you.